This is the walkthrough for the Meiosis Lab, so the cell division lab. You're going to need your cell division lab kit, so it is all going to be in the one Ziploc bag. I've got mine unpacked here. It's going to have the little Ziploc bag of Cheerios. It's going to have a pair of pennies. You're going to have your beads. The beads you can go ahead and set aside. You're not going to need that until we get to the meiosis part. We're going to do mitosis first. So you can set the beads way over out of the way. Just be careful that they don't roll off somewhere. And I already gave you the directions to go ahead and take your pipe cleaners and cut them all in half. So you should now have 4 pieces for each of the lengths. So 2 blue and 2 red for each length that you've got, just like what I've got here. So you should have a total of 16 pieces of pipe cleaner. Now, if you look there in your lab, you can see the pipe cleaners are going to be our chromosomes. The pennies are going to be our centrioles. These are what are going to be used to be the anchors in the cell division. The Cheerios, the cereal, those are going to be our centrosomes. These are going to be what hold the DNA together, the chromosomes together when the DNA replicates. So we'll use those. Now you don't have to use these, especially if you have a gluten allergy, because these are not gluten-free, but they do help if you can use them to illustrate what we're going to be doing. And you also need a piece of paper, so you should have 2 pieces of blank paper folded up in your box. That's what this is for. So what we're going to start off with is, let me get the fuzz off my paper. We're going to start off with, like I said, modeling mitosis. So this is going to be a review of the steps of mitosis. So the first thing we've got our piece of paper. This is going to represent, in this case, a plant cell. The edges are the cell wall and the cell membrane just inside. So we want to first get a circle. So you want to do this with a pencil because we're going to need to be able to erase it later on. So you want to make a pretty decent sized circle inside of your paper. And we're going to start first with interphase. So you can see there what a real plant cell looks like in interphase. What we need to do is make interphase happen using our little pipe cleaners and the cereal. So you're going to need some of those Cheerios. You should have enough. I should have given you extra so that you have enough of these guys. What you're going to do to replicate the chromosomes, if we want, we can show that if we put one of each of these guys in here. So you want to put 1 red and 1 blue of each of the sizes into your cell. So those are the 2 big ones. There's that size. There's that size. There's the red one. Okay. So this would be the cell before DNA replication, right? So single copies of each of the chromosomes. We've got the colors to represent the chromosomes that originally came from mom. So we're going to use red for the ones that originally came from your mom and blue for the ones that originally came from your dad. So the first step is to do DNA replication. So we're going to do that with the Cheerios so that we can keep chromosomes together. So you're just going to match them up with size and color and jam them through the Cheerio and make them into little X's. You don't need to twist them. The Cheerios will hold them together. once you get them all replicated, we're going to place them all in a pile because that's what they would look like in interphase. They'd just be in a big group in the nucleus. You're going to take your pennies and put them in the cell, because those are the centrioles. And now you're going to go ahead and in your sketch sheet, you're going to make a sketch of what their cell right now looks like in interphase. Now you can print off the sketch sheets that I provided, or you can just go ahead and sketch this in a box on a sheet of paper and then just turn in all of your sketches on Pantherloom. Now we're going to move into mitosis. So you've got your cell now finished in interphase. So these centrioles, they're just kind of hanging out. In prophase, we're going to do a couple of different things. So remember, prophase is kind of a busy stage in mitosis. So the centrioles are going to start moving towards opposite sides of the cell. The spindle fibers start to appear, so you can start drawing little lines off of these guys. If you don't want to draw those lines in right now, that's okay, because you might have to erase them to move your centrioles again. The nuclear membrane is going to break apart, so to show that, you're going to start erasing your nuclear membrane. This is why you need to do it with a pencil. You can also do it with an erasable pen if you have one. I know not all of us own one of those. All right, so nuclear membrane's breaking down. The chromosomes condense, so they start to be visible as more structure, so you can kind of show that by spacing them out a little bit more. They stay in the nucleus, but they're more visible. And that's the end of prophase. So you can start to see what you're going to be doing. So I'm going to let you do metaphase. So you can see there the procedure for metaphase. So see steps for metaphase, what's going to happen. So you're going to go ahead and do the procedure of putting those chromosomes in the center of the cell. And for both prophase and when you finish prophase and before you move to metaphase, and then when you're done with metaphase, make sure to make sketches of what your layout looks like for both of those. So I'm going to go ahead, I'm going to show you what anaphase looks like as far as what you're going to do for that part. So you're going to have your chromosomes lined up. You've got your pennies. Your chromosomes are going to be lined up in the cell. Now, you may have noticed that it's kind of hard to line them up this way. If that's a problem, you can line them up long way and put your pennies on the sides of the paper. That will give you more room. All right. So this is what your metaphase should look like without the nuclear membrane. And you should have lines going from the penny to each of your chromosomes on each side. Now for anaphase, anaphase, remember, is that stage where the chromosomes are going to get pulled apart, right? You're going to pull one chromosome or one chromatid to each side. So the way you're going to do this is you're going to snap or crush that piece of cereal. So you can do that just by snapping it like that, and then moving the chromosomes to each side. All right. If you want, you can eat the cereal. They're probably pretty stale by now. They've been in your bags and boxes for several weeks. So you do that with each of your chromosomes. So snap them and pull them. If you want to avoid maybe a little bit of a mess, you don't necessarily have to snap them. But that's actually what happens. So that's why I put that in there, so you can get the idea that these are actually going to break and pull apart. So you're going to do that to all of your chromosomes, put them in the correct place. So they're going to all get pulled apart. So you'll have them half on each side. Make your sketch of what anaphase looks like at the end of the steps. I'm going to fast forward. So, oh, by the way, if you want to avoid the mess, like I said, you can just slide these guys off and put your chromosomes down. Okay, so I finished Anaphase, and now we're going to move into Telophase. So Telophase, as you can see there, if you remember, that's when the chromosomes are going to unwind. So they're going to pile back in together. This is also where the nuclear membrane comes back. So you're going to, you can move your pennies out. You're going to bring in the nuclear membrane, right? So you can erase the other parts that you've got there. Make your sketch of what you've got now. And then you're going to do cytokinesis. So you're going to draw in lines to split your cell, and then you need to take a picture of your complete set of new cells. So with the chromosomes there, the new cell walls, those are the new those two lines that are down the middle. I'm going to do the Meiosis Lab Procedure now. And what we're going to be doing, we're going to use the same set. So you can see I've got a new piece of paper, got the nucleus drawn in. So we're going to start with that as well. Now we're going to get one of each color of each of our chromosomes, right? And put them in. You also are going to need your beads, right? So go ahead, grab your beads, and you're going to need your Cheerios again. So grab your Cheerios. Now before we go any farther, you want to go ahead and answer questions 1 through 3 right now. So right now you do have a diploid cell, because remember, diploid means two copies of the chromosome. You have a copy that was inherited from your mom and your dad. So we're still going to do the same thing we did with mitosis. The blue ones are going to be dad, the red ones are going to be mom. Now the colors are going to be important. Now we're going to take all of our pipe cleaners and we're going to do DNA replication just like we did with mitosis. because we're in interphase. So go ahead and put your chromosomes together again with those little pieces of cereal, matching them up by size and color. Okay. Now, we're going to do something a little different at this point. This is where you're going to use the beads. You're going to take the beads and on one leg of each of your chromosomes, you're going to put on a matching bead. Okay. These beads are representing genes on the chromosome. So blue for the blue and pink for the red. I know the red and the pink don't really match. I didn't have any red beads. All right, so you should now have all of your chromosomes copied, and we should have markers for the genes on one leg of each of the chromosomes. So now we've got our pile of chromosomes. We're going to put our centrioles back in the cell, and you're going to answer questions 4 through 6 on your worksheet and sketch what the cell looks like right now. And again, you can, if you've printed off the sketch sheets, then you can just draw them there in the box, or you can draw them on a separate piece of paper and submit pictures of that along with your question sheet. Now we're going to move into the stages of meiosis, because this is interphase in meiosis. So we're going to do prophase 1. So prophase 1, we're going to do the same steps that you did in mitosis. So those centrioles are going to start moving towards the center. Now, you can either move them so they're going towards here, or move them so they're going towards here. It's up to you. The nucleus starts to break down, so you can erase parts of the nucleus. The chromosomes have condensed, so they're easier to see, and the way we can do that this time is we can put them together as their sets, because that's actually what they do in meiosis. Remember, they get together there in their partners, right? Called those homologous chromosome pairs. So you can set them up so that they're in those pairs. And now, before we go any farther, we still have one last thing to do, which is the crossing over. Because remember, in prophase 1, we have to do crossing over. So the way you're going to do crossing over with our pipe cleaners here is you're going to take the pink bead off of the red chromosome and the blue bead off of the blue chromosome, and you're going to just swap them. Okay? And you just do that for all of them. So this shows that we're changing that little part of the genetic information on the chromosomes. So that blue gene is moving to the red one, and the red gene is moving to the blue one. Right? And you're going to do that for each homologous set. So we're moving one gene for each set. So that's what the second step on your prophase 1 procedure is telling you to do, is to swap the beads just like you saw me do here. Then you're going to move to Metaphase 1. And this is where the groups, the sets of homologous chromosomes are going to form a line in the middle. Okay? So you've got your centrioles that have moved towards the opposite sides of the cell. These guys, remember, move together as sets. So they are going to stay together as those pairs, just like that. Okay. And remember, when this happens, they don't all line up so that all of Dad's chromosomes, all of the paternal chromosomes are on one side and all of Mom's or the maternal chromosomes are on the other. So you should have some sort of mix, and they don't have to be just like mine, right? So you can have them set like this or like that, right? There's lots of different combinations of how they get set up. But you should not have something like this, because that's very, very rare to have happen. That all of the ones that you inherited from Dad are on the same side as all the ones you inherited from Mom. Okay? So make sure that they are mixed up. Once you have them lined up, you should have a spindle fiber coming from each side. So one from each penny to each side, and then we call that a tetrad. Tetrad for 4 chromosomes, pair of homologous chromosomes. Make sure to sketch Metaphase 1 in your worksheet, and there are a couple of questions there between Prophase 1 and Interphase. Now we're going to do Anaphase 1. Anaphase 1, remember, is you're going to separate the homologous chromosomes. So that means you're just going to pull these guys to either side of the cell. So these spindle fibers would pull on those. So you're going to do that to all of your sets. Once you've done that, make sure to sketch Anaphase 1. And then go ahead, do Telophase 1, which remember is the same as Telophase and Mitosis. So you're going to get the nucleus. You're going to put in your nucleus. You're going to take out all the spindle fibers. Then make sure to sketch Telophase 1 for both of your cells and answer the questions that you've got there on your worksheet. And you'll see that on your worksheet, I've got Telophase 1. It's also Telophase 1 and Cytokinesis, so you can go ahead and draw your new cell walls. So you're going to divide your paper in half, just like we did for Mitosis, because we've got 2 new cells. All right, so we have now made it to interphase between Meiosis I and Meiosis II. And nothing is going to happen right now. So I'm going to answer number 14 on your worksheet. We're going to head now into Meiosis II. This is the same steps that we have in mitosis. It's just we're going to be having it happen at the same time in 2 different cells, right? So prophase 1, you're going to have the nuclear membrane breaking down. Your chromosomes are going to condense, so they'll be easier to see. Now I'm just going to do it in 1/2, right? But remember, you have to do it in both. So once you finish prophase, you can sketch it. For this part, you can leave out the centrioles, the pennies, just because it gets a little hard to maneuver them around. But remember that they'd be there, so just sketch your spindle fibers out to a spot where they would be. So then metaphase, these guys are going to line up in the middle, right? And it's okay if they're piled on top of each other. Then anaphase, you're going to break the Cheerios and pull them apart. Lost a gene. There we go. That was on that one. All right. So anaphase, then telophase. Where we've got our little groups of chromosomes with the nuclear membranes coming back. And then cytokinesis with the new cell walls. And so here you can see I've got 2 of our 4 for mine. And you can see just from these 2 that we have different combinations of chromosomes. So they do both have blue and red for these 2. But the genes are different, right? So this one, this is the same chromosome, but this one has that blue gene on it. This one is the same chromosome, but it has a pink gene on it. This one's the same chromosome, has a pink gene on it. Right? This one has a pink gene on it, no gene. So I think though something happened on that one's supposed to have a blue gene. I think it might not have gotten swapped. So that is the walkthrough for the mitosis and meiosis. When you get to the end of Telophase 2 and then cytokinesis, make sure to take a picture of your 4 completed cells and put that in with your worksheet and answer those final questions in your worksheet. If you have any questions, let me know.